Because this is the one where we just go from prophase to metaphase.
Okay.
We just totally skip
Prometaphase.
Prometaphase, yeah. You really kind of just start the same.
So you have a bunch of condensed chromosomes inside the nucleus, inside the cell.
Yeah.
And then on the side it has the spindles.
Yeah, you're kind of starting to get, a little more. I'll try to draw them a little thicker. They're really not really condensed, but they're kind of condensed,
Condensed-ish.
Yeah, they're getting there. And then during prophase, while this is all happening, they start to condense more and more and more.
During what?
During prophase?
Yeah.
And during this time, during prophase, this is when there's kind of a lot of things going on. We have crossing over that occurs.
Mm-hmm.
So the kind of, this chromosome goes into that one and whatnot, and this chromosome kind of goes into this one, and they kind of cross. Do you remember crossing over kind of?
I'm confused. So that's still in prophase?
So yep, this is also in prophase.
What exactly is crossing over? Is it because you have genes from two people?
Yeah.
So that's what's crossing over?
So, yep. So you have the genes from you have these two homologous chromosomes.
Mm-hmm.
And this one, I should have drawn them closer. This one kind of leaks over to here.
Mm-hmm.
And then, this chromosome right here might leak kind of over into here. And it kind of just mixes. So then in the end, it comes out looking like...
Like it's just intertwined, basically?
Yeah, kind of. So you're like this.
Mm-hmm.
And you may be like... Some has some of one now, and then some of the rest. It mixes them up.
Yeah, okay.
And then the other one would just be the opposite of this, so it would look like...
But they won't necessarily be like 50-50.
No, it's not always a set ratio or anything.
Like it can be, but
Yeah.
it won't necessarily be.
Yep. And so prophase, there's a lot going on, and so it kind of depends on what moment in time you're talking about prophase, but it goes from this practically to really condensing them,
Mm-hmm.
to having crossing over happen, so these kind of get mixed up. And then the thing that holds these chromosomes together once you get to this spot are called chiasmata. It's like where they crossed over, and now since they're still like connected, it holds them together.
Wait, how are they held together?
By these remnants of crossing over. So kind of like right here in this picture. See, you can see where one crossed over to the other.
Mm-hmm.
And they're still connected, so that's what's holding them together.
So the fact that they are connected, that's what's holding them.
Yeah, that's what's holding them there together.
Okay.
Yep. Before crossing over, they're just held together by the synaptomal complex, which is just a bunch of proteins that hold them together. So we kind of look like this, actually. We start off like this in prophase, and we work our way having these things kind of held together.
Is it okay if I take a picture of that?
You're fine, yeah. And these mitotic spindles are still starting to come together. They're not fully there yet. And even to like right before we get to where it's like Because in the last one, if you remember, we had prometaphase where they just attached.
Yeah.
Well, now we just consider that a part of prophase. So they're not on the metaphase plate, right?
So what it's showing is that they're like starting to line up.
So they're attaching and they're on their way to line up.
Okay.
But they haven't. They're not lined up. So that's all. A lot happens during prophase during meiosis 1.
Okay.
All this condenses, starting to work. You have them condense more. You have crossing over occur. After crossing over, you have these things attach, and you have them attached to the mitotic spindles, and they're starting to kind of get to line up. But they're not lined up yet until metaphase. No. That they're lined up.
And so metaphase is the same in meiosis and mitosis.
Yep, they're just lined up
Basically.
there. And you just kind of look, they're all along that line. But the difference that we look for here is now, instead of just having all the chromosomes lined up like this, we have the homologous pairs lined up across from each other, which is important to make sure.
Which is different from mitosis.
Yep.